Okay, so welcome. This video will be on beta oxidation of fatty acids. So fatty acids are beta oxidized actually in most cases during fasting, like when the glucose level in the body has reduced. Actually, when the carbohydrates in the body have reduced, so that means the supply for glucose has reduced, and this time the body will return to fatty acids. So fatty acids, they are normally stored as tags. They are stored as tags within the adipose tissue. So during starvation, these tags will be broken down by lipolysis. They will be broken down by lipolysis. And so this lipolysis, how will it occur? So it will occur by, actually this time now let's first talk about lipolysis. This lipolysis, we shall have a tag. A tag is actually a glycerol molecule, glycerol backbone, but then this glycerol backbone is connected to 3 fatty acids. So that is fatty acid number 1, fatty acid number 2, and then fatty acid number 3. So this is number 1, number 2, number 3, and this is the glycerol backbone. So this is what we call the triacylglyceral, and this is how fatty acids are stored in the adipose tissue and in the muscles. So during lipolysis, we first utilize the first enzyme, which is called the adipose triglyceride lipase. So adipose triglyceride lipase will first break down the bond holding the first fatty acid to the glycerol backbone. So this one will be broken down by adipose triglyceride lipase enzyme. This is the enzyme found in adipose tissue. So it will first break down the first fatty acid by addition of water. So it is broken down by hydrolysis. So by addition of water molecule, we shall have the breakdown of this fatty acid from the glycerol backbone. And so that means after removal of the first fatty acid, we shall remain with our glycerol, but this time connected to fatty acid number 2 and fatty acid number 3. So that means this time what we have is what we term as DAG. This is diacylglycerol with 2 fatty acids. So the next enzyme would be, which is the rate limiting enzyme for the reaction, and it is called hormone sensitive lipase. So this hormone sensitive lipase will now catalyze the breakdown of the second fatty acid from the glycerol molecule, and this second fatty acid, this second fatty acid is normally unsaturated fatty acids. The first and the third, they are always saturated, then the second is always unsaturated fatty acid. So now this hormone sensitive lipase, which is the hormone rate limiting enzyme in the lipolysis reaction, that means we shall have this hormone sensitive lipase will catalyze by hydrolysis, the hydrolysis reaction. Hydrolysis means it is done by addition of water, and so it will catalyze the breakdown of the second fatty acid from the glycerol backbone, and so we shall remain with glycerol and fatty acid number 3. So we shall remain with this, which is termed as a monoacylglycerol, MAG. So it is a monoacylglycerol. Now this one, now what we shall utilize the next enzyme or the third enzyme, which is monoglyceride lipase. So this monoglyceride lipase enzyme will catalyze now the breakdown of this bond holding the glycerol molecule to the third fatty acid, and so we shall remain with glycerol plus our three fatty acids, which are now free. These are now free fatty acids. They can go into the saturation. So that is lipolysis. Our main emphasis was to break down the tags to release the glycerol backbone and the three fatty acids. And I said the first reaction, the first fatty acid, its breakdown is catalyzed by adipose triglyceride lipase. Then the second fatty acid, its breakdown is catalyzed by hormone sensitive lipase, which is the rate limiting enzyme for the reaction. Then the third fatty acid, its breakdown will be catalyzed by monoglyceride lipase, and so we shall have our glycerol and three fatty acids. This glycerol, its fate, it can either be combined with other fatty acids during lipogenesis to form more tags, or it can be converted to diacyl, it can be dihydroxyacetone phosphate. This is an intermediate during glycolysis. We shall talk about it during when we are talking about gluconeogenesis, but that is one of the fates. So glycerol can be converted to dihydroxyacetone phosphate, which is an intermediate during glycolysis. Then these three fatty acids, now they can go to beta oxidation. So beta oxidation, which we are also going to describe. Actually, from here I'm now going to describe beta oxidation. Thank you for watching. Subscribe. So the next video will be beta oxidation.